All too often, we focus on the movers and the shakers of the proteinaceous world. The best binding affinity, the highest catalytic activity. But what about the proteins whose job it is to just stand around? These are the unsung cytoplasmic heroes, the structural proteins. One goes particularly unnoticed in the postsynaptic densities of neurons, PSD-95. Standing for postsynaptic density 95, PSD-95 silently works to ensure that certain receptor-mediated ion channels, namely NMDA and AMPA receptors, are in proximity to one another to create coordinated neural depolarizations. PSD-95 is by no means an overly complex protein. It consists of 3 PDZ domains, a single SH3 domain, and an inactive guanylate kinase domain linked by disordered, unstructured linkers. For the sake of time, only the PDZ and SH3 domains, which bind proteins and provide functionality, will be discussed. The PDZ domains are the business ends of PSD-95, performing most of the protein interactions necessary to provide structural functionality. It is comprised of a beta-barrel S structure of antiparallel beta sheets along with 2 flanking alpha helices. The primary binding site of the PDZ domain, responsible for sequestering the C-termini of target proteins, is shown here, comprised of positively charged residues which form strong interactions with the relatively negative C-terminus. The interacting residues are shown here, with the PDZ domain of PSD-95 shown in color, and an example peptide being shown in gray. The next domain of note is the SH3 domain, which as shown here is composed of several antiparallel beta sheets forming a barrel, which interacts with highly prolinaceous segments of peptides in a hydrophobic binding pocket, shown here, where blue regions show hydrophobicity. A closer view of the interactions can be seen here with an example peptide in red and the SH3 domain in green. However, at this point, many of you may be wondering what PSD-95 actually does. As stated before, its main role is to be a scaffold for certain large ion channels, but why is this important in the first place? Couldn't the channels make their way to the postsynaptic membrane and lodge by themselves? Theoretically, yes. However, the NMDA and AMPA receptors form a very special pair that require proximity to work properly. Both the AMPA and the NMDA receptor bind glutamate to allow for the influx of sodium and calcium into the neuron. However, their sensitivities and properties are extremely different. Firstly, AMPA receptors open and close relatively quickly upon the binding of glutamate, creating small areas of depolarization around themselves. NMDA receptors, however, stay open for much longer and provide a steady influx of calcium ions. Also, whereas AMPA receptors bind with a low affinity, being in the micromolar range, NMDA receptors bind glutamate with nearly 1,000-fold higher affinity in the nanomolar range. So why would the sensitivity of NMDA be so much less? This property is a result of the NMDA channel having a magnesium plug, or rather a magnesium ion lodged within its center. This stops the influx of any other ions until there is a depolarization from inside the cell. This is where AMPA becomes particularly important. If AMPA is activated in close proximity to an NMDA receptor, there is a high probability that the NMDA receptor will have bound glutamate and is simply waiting for the magnesium plug to be released. The small areas of depolarization from the AMPA receptors facilitates this release, allowing for the NMDA receptors to evoke long-lasting effects in the cell. Because of this need for proximity, AMPA and NMDA receptor coupling is a highly regulated system that relies on proteins such as PSD-95 and others to facilitate the specific binding of C-termini and the subsequent localization of these receptors. The specific residues in each of the PDZ domains, as well as the SH3 domain, have evolved to form preferential interactions with the C-termini of not only these receptors, but other scaffolding proteins, creating a complete and stable postsynaptic environment. However, sometimes this postsynaptic density needs to be pliable enough to adapt to the needs of the brain and the neural environment, namely in times where the strength of the synapse is altered. PSD-95 is a central player in this process, facilitating something called long-term potentiation, or LTP. This process relies upon a strong glutamate signal activating enough NMDA channels to create a heavy influx of calcium ions, which activates many calmodulin-dependent kinases. The ultimate downstream effect of this strong depolarization is that more AMPA receptors are brought to the synapse from where the signal was received. This allows for future signals to more easily depolarize the neuron, strengthening the synapse. However, the additional NMDA and AMPA receptors that are brought to the synapse have to be added to the existing scaffold, which is where PSD-95 becomes essential. In mice where PSD-95 has been knocked out, synaptic strength was shown to be significantly decreased, most likely due to the lack of postsynaptic NMDA and AMPA receptors being placed after the old ones turn over. The synaptic plasticity shown by learning and memory tasks was also shown to be at disadvantage in the absence of PSD-95. The ability of neurons to form more meaningful, stronger connections across time, the physical result of long-term potentiation, was also hindered, with neurons forming less dendritic spines and having deformities in the spines as well.